So good afternoon, everyone. Thank you very much for joining. My name is Udbhav Ram. I'm a first-year undergraduate student here at Mcmaster, and today I'll be presenting my research on an augmented reality setup to improve accuracy in surgical chronic kidney disease in mice. This is a great collaboration between us here at McMaster University, as well as our colleagues at St. Joseph's Healthcare Hamilton and the Hamilton Center for Kidney Research. So before we dive into exactly what our research is, it's pretty important to understand what chronic kidney disease is. So this is known by many names, just kidney disease or kidney failure, but really it's characterized by the loss of kidney filtration over some period of time. So in this case, we can see it's usually characterized in 5 stages, going from stage 1, which is a perfectly normally functioning kidney, all the way to stage 5, where we have complete kidney failure. And you may think that this isn't something that affects you or anyone you know. But I can tell you that more than 1 in every 7 American adults suffers from chronic kidney disease. That's more than 37 million people in the US alone. So it is very much something that we need to solve. And I say that because chronic kidney disease currently has no cure. And that's crazy. 37 million people in the US alone and no cure. So as researchers, it's important to understand the best treatment pathways. And in order to do this, we need to find something that's analogous to losing this filtration. So what is the best way of simulating this loss of filtration? Now this brings me to the 5/6 nephrectomy. In a word, the 5/6 nephrectomy is a removal of 5/6 of the kidney mass in order to simulate this loss of filtration over time. And of course we do this in mice models because it's not feasible to do this in humans. And the first step is to remove 2/3 of the left kidney of a mouse. Following this, about 1 week later, we remove the entire right kidney. So by the end, we've removed about 5/6 of the total kidney mass. And here we want to aim to have our left kidney be resected at a mass ratio of 0.6. And I'll explain why this is important in the next slide. Now what we can see here is data collected by all of our operators who have performed this procedure over many, many months. And as we can see, the important 2 lines are 0.5 and 0.72. Now the reason we've chosen these numbers is not arbitrary. If we resect any less than 0.5 of the tissue of the left kidney, what we end up having is not enough data to produce meaningful results about that kidney or its filtration. And if we resect any more than 0.72, we greatly increase the risk that the mice will actually die. So it's very important that our operators stay within these 2 limits. However, as we can see, there's a great spread in actually how much they've actually resected. And so all of these red dots that you can see are mice that either died or were rejected from our study due to operator variability alone. And this is the problem that we are trying to solve. So our current approach is going from the kidney you can see right up here, where we have nothing on it, and this can be a picture or a video, and going all the way to this kidney over here, where we have nice lines that the surgeon can then operate upon. And we do this using computer vision, mathematical modeling, and augmented reality in order to perform an efficient analysis of the kidney and tell the operator where to cut. Now, as far as our current architecture goes, for hardware, we have 2 things. We have a computer, and as you can see here, it has some specifications, but this is just to say it's not a supercomputer. It's not something that you're not going to find at any lab or any home setup. What you can also see is an enclosure. This enclosure includes a high-resolution camera and some lighting, and it mainly serves us 2 purposes. One is to mitigate shadows and occlusion and to control the light very efficiently. The other is to create a mobile and self-contained platform that we can take from our test bench to a surgical environment and anywhere else that we might want to test. Now, this brings us to our current working application. As we can see, the algorithm is able to track the kidney throughout the frame and display certain metrics. What it's also able to do is track the kidney through its 360 degrees of rotation. In addition, as we can see on the very bottom with the percent resected, as well as this bar, is the ability for the surgeon to actually change the amount resected during the surgery itself. And this happens live, frame over frame. So the first step in actually achieving an algorithm like this is camera calibration. And you might be wondering what this is right here. And this is actually what helps us with camera calibration. So the question we want to answer is how to standardize or normalize the objects in our scene. We know the pixel length, as we can see with this red arrow right here. And what we lack are SI units for this kidney. So this could be 10 millimeters or 10 centimeters, and we just don't know. So what we're able to do is to convert and calculate this SI unit measurement using our Aruka marker, which is the black box here. As I mentioned before, this is an Aruka marker, and we use it commonly in computer vision and augmented reality in order to get scale and orientation parameters for different objects. So it's known as a fiducial or reference object because we always know the qualities of this object. And it helps us to create a pixel to SI unit conversion. For example, we can measure it to be 10 pixels in this picture. And what we can then do is, because we know the exact centimeter amount, is create a pixel to centimeter ratio. This then helps us to convert the kidney into an SI unit, in this case 2 centimeters, and we can then proceed with the analysis. So now that we know exactly how our system is standardized and we know all the object's lengths, what we then need to know is where our object is and how it's oriented. In this case, our object detection algorithm is fairly simplistic because we have a white background and a fairly contrasted image. So there's no need for anything fancy here. All we do is we have a binarization algorithm which uses specifically Otsu's method of thresholding in order to pick out the most prominent object in the scene. In this case, that's our kidney, and it's fairly easy to see. Now the next question is orientation, and you might be asking, well, why do we need to know orientation? Well, as you can see here, with our blue kidney and our orange kidney, these are the exact same model. However, the blue kidney is tilted at 45 degrees. And as you can see, both of these boxes have wildly different scale parameters. So across 360 degrees of rotation, we'll have 360 different scale parameters output. And this, of course, is not ideal. So what we've done to solve this problem is using something called a minimum area bounded rectangle. And that's what you can see here in green. What it allows us to do is output a constant scale parameter across 360 degrees of rotation, which will allow us to perform a more efficient calculation. What this also gives us is an insight into the orientation vectors, which allow us to quantify the actual orientation of our object and use it in our later analysis. So now we've covered everything about 2 dimensions. We know where our object is and we know how it's oriented. The next question is, how do we know about our object in 3 dimensions? Because at the end of the day, we want to perform a volumetric calculation. So what we've done is had MRI experiments using a setup that looks something like this, where we have an Eppendorf tube, we have 2 kidneys that were transported and stored ex vivo in formalin, and the results look something like this. As you can see, you have an MRI, this is fairly standard, where we're going through the object on one axis. Special thanks to Manoj Singh and the Mcmaster Animal Facility for having enabled us to get this MRI data. And so the question is, well, how can we get a usable 3D model for us to actually use in our experiments? So as we know with any video, and especially MRIs, it's just a series of static images. So what we can do with these raw 2D images is assign each a thickness and then stack them one on top of the other iteratively until we reach a model that looks something like this. What we can then do with this model, and why it helps us, is we can re-slice it back into separate slices which have a constant depth, and this will then help us in the next steps. So now, in order to use this 3D model to actually predict where our cut points are, we need to do a couple of things. So we know that our volume for any given frame, in this case we're taking a keyframe, is given by product of area and depth. So we know the area is, of course, the number of white pixels in this frame, or it can be inferred to be the number of white pixels in the frame. And we know that the depth per slice is constant because we're the ones who sliced it. So what this gives us is that we can calculate a volume for every single frame, and not only that, we can calculate a volume for any point along a certain axis. Why this helps us is because we can go from our 0% volume to a pre-calculated 30% volume and iteratively add up our volumes until we get to that point. Once we're there, we stop and we indicate that here's where our cut point should be. Now, illustrating this to the surgeon is a matter of overlaying this model along with its cut points onto the live feed, along with the correct orientation and scaling parameters. So our current limitations and our next steps essentially include a modification because our current setup does not include the intricacies of a surgical setting. As I've discussed before, the current setup has a white background and a well-contrasted kidney. However, what we can see here in our surgical setting is a variety of other objects that may clutter the scene. And with the current algorithm, as we can see here, the binarized image is much too cluttered. There are no meaningful extractions that we can make from this image. So our next steps, as far as software goes, will likely be a deep learning algorithm that will be able to instance segment the image based on various things, and will be able to tell us exactly where the kidney is without worrying about any of the clutter. Now our next iteration on hardware will likely be something like this render. As we can see here, we have the surgeon who has a tablet just over their hands and a lighting and camera setup that is directly underneath. And what this allows the surgeon to do is to complete the surgery with our augmented reality results enhancing their view, and not only that, to do it without actually ever seeing their hands. Their only view of their hands is through the actual system itself. So in summary, there's an increased risk of death or reject in mice due to operator variability alone. We have developed a prototype with a laptop and attached webcam enclosure, as well as software that is able to detect a kidney in a video. We've done mathematical modeling of a kidney based on ex vivo MRI data, and we have real-time prediction of incision sites frame over frame. Finally, we're able to bring all of this together to the surgeon in a user-friendly manner using augmented reality to provide an enhanced view. Finally, I'd like to thank all of my collaborators, Dr. Michael Reinstatter, Dr. Sebastian Himbert, Dr. Joan Krupinski, Asfia Sumro, Dr. Timothy Ryan, as well as all of our funding agencies for having enabled this research. Thank you very much for joining.